So we left off, we were looking at homeotic genes, specifically genes in a cluster called HOMHOCs, which specify the identity of segments in the development of a fruit fly. One in particular, UBX, if you knock it out, what UBX does is cause all of the abdomen of a developing fly to turn into a chain of thoracic segments. That's what UBX does if you knock it out. So UBX's function is basically to ensure that the rear segments of the fruit fly develop into abdominal segments, and in particular, don't sprout wings and legs. This is what UBX does in insects. It doesn't do this in crustaceans, which we think include the ancestors of insects. So somewhere this one gene has shifted function just a bit, causing insects to no longer develop legs on their abdomen. Did they used to have legs on their abdomen? I'm glad you asked. We think all arthropods evolved from something that had legs all over its body. This is a 510 million year old fossil that looks a little bit like a caterpillar. Normally, fossils of squishy organisms don't get preserved at all because they just rot before they can leave any impression in sediments. But this one comes from a very special and magical place called the Burgess Shale up in the Canadian Rockies, where critters were buried fast enough that you can find impressions of their soft tissues. And this thing called Aschia would have looked a little bit like a caterpillar. The actual size is maybe an inch long, roughly 2 to 3 centimeters. And it has a bunch of appendages, right? You can see a bunch of legs on it. You've got this elongated body. Dang it. This is the elongated body. And then here you've got a bunch of legs. And they all pretty much look the same, except for one pair of legs that's actually looks the same as the others, but it's facing forward. Something like this, although not exactly Aschia itself, is thought to be what the common ancestor of arthropods would have looked like. And starting from something like that relatively simple fossil, something like this thing labeled onychophoran up here, what we think has gone on is that body plans of the arthropods, that's chelicerates, which includes your spiders and scorpions, crustaceans, which is crabs and lobsters and roly-polies and sea monkeys, myriapods, which is your centipedes and millipedes, and insects have evolved from something like this very simple fossil up here by specializing, enlarging, and reducing segments and body parts. It's basically exaptation gone wild. Specifically, we think that insects are derived from a centipede-like ancestor. And what's happened is that what used to be walking legs got modified into antennae and mouth parts and walking legs. And it's not shown clearly here, but genitalia of insects, by the way, which are usually kept kind of tucked inside, are thought to be modified legs as well. So you start with something that's got legs on every segment, and then some of the segments get shifted around, modified, clustered together, and the appendages on them get reworked for very different purposes. So it's exaptation gone crazy here. Lots of it going on. To make an insect out of a centipede-like ancestor, this thing labeled myriapod down here, UBX evolved to suppress genes that act in the thorax notably a Hox gene that promotes limb development called DLL. UBX essentially says, whatever you're doing ahead of the second thoracic, don't do it back here. UBX blocks DLL. DLL promotes leg development. UBX stops DLL. So that's how UBX keeps insects from developing legs on the abdomen. And in fact, we can get some insight into how this evolved, because if you look at the fossil record of insects, some very early ones, this is actually a fossil from another location where soft and squishy things sometimes do get preserved, a place called called Mason Creek in northern Illinois. This is a very early fossil insect, and it actually did have legs on its abdomen. So UBX apparently was not completely suppressing legs on the abdomen here, because there's still little vestigial leglets here on the abdomen. UBX is not completely blocking leg formation. Meanwhile, we don't know a lot about the evolution of insect wings. We've got some gaps in the fossil record there, but what we think is going on is that there's a little plate right here, almost like a shoulder pad, that's originally part of the leg joint. It's labeled pleuron up here, or SCX. I forget what that stands for. But that's originally a part of the joint, but we think it grew larger and eventually gave rise to the wings. This is another case of exaptation. You're taking a structure that's already present and converting it into something very new. And so here we've got some more insects. This is another fossil insect called Dasyleptus, and you can see it's also got little leglets on the abdomen. Epiromymus right here, this is an early fossil mayfly, and it's a larva, and it would have legs on its abdomen, although you don't see them in this view, but it also had wings on the abdomen. So remember that in fruit flies, UBX says, don't put legs in the abdomen. UBX, in this case, in these fossils, is not fully on the ball yet, is not completely doing its job, because these early insects are developing little leglets on the abdomen, and in some cases, they're developing winglets on the abdomen as well. This is what you would expect if UBX is not fully functional. So the shift from something with legs on the abdomen, like a shrimp, to something with no legs on the abdomen, like an insect, involves some kind of change in the way UBX works. And you can see intermediate stages in that process in the fossil record here. This is cool. We are putting together fossils and DNA into a coherent story. Notice again that wings evolved, we think, from a part of the leg joint, which means that wings and legs ought to go together. And in fact, one of the earliest known winged insects actually has not two pairs of wings, but three. There's the first up there. And then there, of course, is the second. And there's the third. And there's little winglets down the side of the abdomen. Right there. So its UBX is not fully repressing development of appendages on the abdomen, because it's got little winglets on its abdomen. And also its thorax is composed of three segments, like all insects is. And every segment of the thorax has a pair of legs, and it's got a pair of wings. And this is the kind of thing we would expect if wings evolve from a piece of the leg. That legs and wings ought to go together in this way. So there, I'm going to leave that story there. What I want you to take from it is this. I'm not going to test you on minutiae of the function of ultrabithorax. What I really want you to see is that there's a fairly small set of genes that acts as a toolkit in animal development. And you can trace some pretty major shifts, like the shift between crustacean ancestors and insects, by looking at the ways in which those genes have changed their function. You can start looking at transitions from one, you know, class of organisms to another in this very nuts and bolts kind of way. You can identify specific genes that are responsible for the changes that we see, and you can start asking questions as to how they changed. And it turns out this is much more than just about insects, because Hox genes are found in all animals. The number of Hox genes is very roughly back of the envelope correlated with how complex a body is. Fruit flies have 9 Hox genes in their HOX complex. Jellyfish have 3. You've got 38. So it's really hard to quantify this, but your body, however you judge it, is probably a bit more complicated than a fruit fly's body. You've got more organs, they're connected to each other in more ways. And this is connected, again, very loosely, to the fact that you've got more Hox genes than they do. You've got more tools in your toolkit. In fact, there are UBX homolog genes that are found in mammals. In mice, they're concerned with early development of the nervous system. Later in development, we know they're expressed in kidneys and gonads and bone marrow and things like that. So UBX seems to lay down part of your nervous system and later on gets involved in building organs. Um, there's actually a mammal homolog that's expressed at a high rate in certain cancers. If you think of cancer as a disease of development gone horribly wrong, understanding how these genes work together is actually going to provide some insight into cancer causes and treatments, maybe, possibly. And here's the thing, how did you get 38 of these genes when fruit flies have 9? Remember that when there's unequal crossing over in meiosis, sorry, or sometimes when you've got polyploidy, which is rare in animals but does happen, you get extra sets of genes. You get gene duplication. Vertebrates, in fact, have 4 Hox clusters, not just 1. We think that somewhere in the transition from invertebrate to fish, there was a complete duplication of the genome, and it may have happened a second time, meaning that early vertebrates had 4 times as many genes as their ancestors did. What this means is that a mutation in one of those genes doesn't necessarily affect the organism because it's got other copies of that gene that can be functional. So you can get, some of these genes are free to evolve and possibly to specialize if you have gene duplication like this. Even before vertebrates had evolved, the Hox cluster duplicated and produced a cluster called parahox, which is also important in development. And the evolution of it looks like this. Down there below, you've got cnidarian, that's jellyfish, basically. You can see they've got 3 of these Hox genes. Flatworms have 4, leeches have 5, Drosophila's got 9. Amphioxus is technically an invertebrate, but it's in our phylum, Chordata. It is similar arm wave, arm wave, arm wave to the common ancestor of vertebrates, and it's got 10. If you look at mammals, there's our 38. We've got 4 clusters of Hox genes because somewhere in the ancestry of vertebrates, that Hox cluster was duplicated. Every one of these clusters is missing a few members, probably, either cut out by a deletion mutation or maybe just lost expression. There's lots of genes that we have that are not expressed anymore. But you can see we've got 38 of these to play with. And if you look carefully at the above diagram, in vertebrates, what an awful lot of these genes do in vertebrate development is lay down the polarity of the nervous system. What parts of that nerve tube are going to become brain, what parts are going to become medulla, what parts are going to be spinal cord, what parts are going to be ganglia, um, and so on. A lot of the molecular machinery that in flies is used to determine where the legs and wings are going to go is used in you to determine where parts of the nervous system go. Um, so there's probably been some exaptation there, but it raises the really cool possibility that research into homeotic genes in fruit flies is ultimately going to pay off in understanding human development better, including certain types of birth defects. Even in very different animals, the same genes turn out to control the development of the same structures. Um, Fruit flies and roundworms look as different from each other as it's possible to be, and yet the same fundamental set of genes inherited from their common ancestor determines where the head's going to go, where the tail's going to go, where's the mouth, where's the butt. Um, the same basic set works in you, and you have the same collinearity that fruit flies have, in that genes that are at the beginning of a cluster tend to be expressed towards the head, and genes at the end of a cluster tend to be expressed in the tail. So you could call the HOMHOCs and other sets of homeotic genes a sort of universal evolutionary toolkit. Ooh, one cool discovery about 20 years ago was there's a different cluster of homeotic genes called PAX. And in particular, there's a gene called PAX6. And PAX6 is involved in eye development. If you have mutations in your PAX6 gene, you tend to develop malformed eyes. So somebody took a mouse PAX6 gene and was able to insert it in cells of a developing Drosophila embryo, and the fly that had thus been treated grew eyes on its legs. So the same genetic switch that says, hey, put an eye here, that is used in vertebrates is also able to work in fruit flies. And in fact, its sequences are very similar to a fruit fly gene called eyeless that we already knew about. So critters as different as a fruit fly and you use the same basic tools to build your bodies, even though the actual shapes of the bodies are very different. Your eye doesn't look anything like a fly's eye, and yet the basic mechanisms that lay your eyes down are very similar. It's strong evidence that we all really do descend from a common ancestor that has passed on this universal evolutionary toolkit to us all. And the evolution of homeotic genes and learning about interactions among homeotic genes is a key part of the way that new phyla evolve on Earth. And I think that's actually where I'm going to stop. So I'm going to stop the recording now.